Okay, guys, welcome back to school. It was an interesting week, not having class for 2 days. I know it was a well-deserved rest. I needed it, so I'm certain you guys did as well. I thought what I would do is kind of go over this presentation one more time, just because we had that hiccup in the week and we lost power, and you guys have exams coming up, so I thought we would just kind of run through all this as an entire presentation, so that way you guys can see it from the beginning to the end, and maybe this lecture will make more sense. So let's look at aerobic respiration, this chapter 3D, and go through it from beginning to end. So as I said earlier this week and later last week, that the aerobic respiration, aerobic metabolism, is basically about electron transferring. So you can see on this slide that this image is kind of representing what the electrons are doing when they're generated through glycolysis and through the Krebs cycle, and being taken off by the NAD plus and the FAD plus. So those coenzymes are really important factors in producing energy and accepting those electrons and delivering them later to the electron transport chain. So the most energy generation really comes down to the passing of electrons through this entire system. I showed you guys this in class earlier this week. This is kind of just a generic representation of everything we've talked about so far. So this would basically be the membrane for the skeletal muscle. This would be cross-sectional view of the fibers in the skeletal muscle. And basically what I'm showing here is that when glucose gets into the cell and undergoes glycolysis to create pyruvate, we really don't have much ATP generation that occurs. So it's kind of a poor system of generating energy initially, and I put here that it's a poor payout. What's important to understand, though, is that in glycolysis, we're not only developing energy, but we're also developing potential energy with these electron carriers, these NAD pluses. So we are pulling off about 6 electrons total in glycolysis, creating these NADH. And those NADH will play a role later on with the electron transport chain in generating actual ATP, like very robust amounts of ATP. We do know that when glucose is converted to pyruvate has essentially 2 potential outcomes. One, it can be metabolized as lactic acid. So it could go this way. If we are exercising anaerobically, the pyruvate will be converted into lactic acid. The hydrogen ion will be released and be transported out of the cell. And lactate will be sent to the liver to basically produce more energy via the Krebs cycle. Pyruvate can also enter the mitochondria. So this right here would represent the mitochondrial membrane. And as we'll see later, there are actually 2 membranes in the mitochondria, but I just did a very generic representation showing that if pyruvate is going to be metabolized aerobically, so if somebody is exercising and they're in aerobic glycolysis, that means that the pyruvate is going down into the mitochondria, where it will be converted into acetyl-CoA and then enter the citric acid cycle. And I identify here that this is kind of a better payoff, because we're going to get a lot more of these NADH and FADH2s, which will help directly generate ATP. So, to kind of go back to some of the things we were talking about in the previous lectures, if we're exercising in a manner where glucose is being converted into pyruvate, and then pyruvate is being converted into lactic acid, then we understand that that is anaerobic glycolysis. So I had showed you guys that graph of the different metabolic pathways, and after we talked about ATP plummeting, and we talked about ATP creatine phosphate system as a buffer system that kind of comes to the declining ATP's aid, the next metabolic pathway would be the anaerobic glycolysis, which would essentially be glucose being converted into pyruvate being converted into lactic acid. Now, when that system becomes exhausted, or we have too much lactic acid and hydrogen ions building up, then we can move into aerobic glycolysis, which would be glucose being converted to pyruvate, and then pyruvate being converted into acetyl-CoA, and acetyl-CoA entering the citric acid cycle. Later on in the lecture, I'll actually do some drawings for you guys, so that you can visually see that and kind of correlate the images together. So on to the next slide. Okay, just a quick recap. When we're talking about NADH being NAD plus being an electron carrier or an electron acceptor, you can see here that the reducing agent will receive the electrons. This is its oxidized form. Once the electrons interact with NAD plus, it then becomes reduced, and we have NADH. And NADH can also donate electrons and be converted back into NAD plus, back into its reduced form. So one thing that we were talking about in class is the relevance and the importance of the NAD plus being recycled. And basically, if you look at this line here, when it donates the electrons, it recycles itself. And there's a couple of systems in the body that actually play an important role in recycling, and we'll talk about that in a moment. So there are essentially two ways. There are more ways than two, but I'm just introducing you guys to two methods in which NADH can be recycled to NAD+. And the first one is the conversion of pyruvate to lactic acid. That recycles NAD+. And then the second one would be at the mitochondria, NADH, donating the hydrogen and an electron into the mitochondria, and basically that will recycle NAD+. And I'll show you guys images of that in a moment so that you can get a clearer picture of that. In this particular one here, this is a highly regulated process where a transporter will basically accept the hydrogen and the electron, and that transporter will take it across the mitochondria membrane. So you can get an appreciation of how regulated this process is to basically allow hydrogens and electrons to get into the mitochondria when they're produced in the cytosol during glycolysis. So the first one here basically shows how pyruvate will be converted into lactate, and how NADH plays a role in that. So we can see that lactate dehydrogenase basically catalyzes that reaction, and we can see that the hydrogens from NADH are put onto the pyruvate molecule, which then converts it into lactate. So you can see the hydrogens being kind of put onto this molecule here. And in that process, we see that NADH is recycled into NAD again. So we always want these electron acceptors available and capable of receiving electrons, because as more glucose comes down through glycolysis, we want these acceptors to be available and not saturated, so that they can receive the electrons and continue generating energy. And then basically I put a little descriptor there that you guys can read in your notes. And basically that the more of this NAD plus that we have circulating, the more of an opportunity we have of generating energy and generating ATP. The next one here is the malate-aspartate shuttle, and this is a really cool system that allows us to take hydrogens and electrons that are generated through glycolysis in the cytosol and basically pass those along into the mitochondria, where they can play a more critical role in generating energy and generating ATP. So we can see here that when we generate NADH in the cell, specifically in the cytosol through glycolysis, this particular molecule can't get into the mitochondria. So there's no transporter to take it across the mitochondria. So in order to be beneficial and helpful in generating ATP, this molecule will donate its hydrogen and its electron, and it will do so through the malate system, the malate-aspartate system. So it will donate its hydrogen and electron to malate, and malate is a specific transporter that is allowed to enter into the mitochondria. So this protein will enter into the mitochondria, carrying the hydrogen and the electron, and then once it gets into the mitochondria, it will then donate that hydrogen and electron to a NADH that is in the mitochondria ready to receive it. So it's kind of a long-winded manner of getting that hydrogen and electron into the mitochondria, but of course, like everything in the body, it's because of the regulation, and it's very important that anything with hydrogen is heavily regulated, because if we have an abundance of hydrogen anywhere in the body, it can alter pH, which will then alter function and potentially form. So essentially what will happen then is malate will be converted into some different proteins here, and then in this form, in the aspartate form, malate can then leave the mitochondria. So gives you an appreciation of how important regulation is. So for the hydrogen to get in, it has to do it through the form of malate. And then for the malate shuttling system to get out, it has to be converted into aspartate, which then can leave. And then aspartate can then be converted back into malate. So very, very cool system. Here is a take-home message. Basically, this is summarizing everything that we've talked about so far. At the end of the previous lectures and at the beginning of this lecture, you guys can read this at your own pace. I just sum it up for you guys. So now we're going to talk about the actual Krebs cycle, or the citric acid cycle, or the TCA cycle. They're all synonymous, and you can see it over here on this side. Pretty complex looking system. We're not going to go that deep into the complexity, though, at this point. If you guys take biochemistry, then we will certainly go into it there. Basically, this is the second major step in oxidative phosphorylation. Because there are passing of electrons in the Krebs cycle, and there is NADH being formed, and NADH being oxidized, this is considered an oxidative phosphorylation pathway. We know that after glucose breaks down, we get this 3-carbon molecule, which we call pyruvate, and you guys can see that glucose is here, and then glucose gets broken down into pyruvate, and then pyruvate dehydrogenase will convert pyruvate into acetyl-CoA, and that is a very important step in this process. And most importantly, the Krebs cycle basically transfers the energy from these molecules to electron carriers. So everything that is occurring here, all the energy that is being extracted from the breakdown of glucose and pyruvate and acetyl-CoA, it's all transferring that energy into these NADHs. So you can see here, you can see FADH2 is being formed. GTP is also being formed, which can be converted into ATP, so you can get one ATP out of one GTP. And if we look at this cycle here, we can see that we get one NADH, two NADH, one ATP, one FADH2, and another NADH. So we get 3 NADH and 1 FADH2. And again, that's potential energy that is ultimately going to go down into the electron transport chain to produce ATP. And this system is important because this is where carbohydrates go when they are metabolized aerobically. So again, if we have glycolysis occurring and pyruvate enters the mitochondria, well, in the mitochondria there's oxygen present, so therefore it's going to be in an aerobic metabolic pathway. And so pyruvate can go in and be metabolized, and we can also get fats and proteins that come into this particular system here to be broken down into potential energy. And you can kind of see that happening here. So again, this system, this Krebs cycle, this citric acid cycle, this TCA cycle, it is in the mitochondria. And basically we can have glucose oxidation, fatty acid oxidation, and protein oxidation that will all occur here. And most important thing to understand is that they all have to be converted into this acetyl-CoA. So this is the particular molecule that is recognized by the Krebs cycle. So if these things are not being converted into what is recognized by the Krebs cycle, then they can't be metabolized and we won't generate that potential energy. So there are some diseases that exist out there, or some hiccups in metabolism, where we don't have this conversion that happens. And if this conversion doesn't happen, then we have a major loss in potential energy, and that can have pretty severe ramifications on systemic physiology. So the Krebs cycle is happening in the mitochondrial matrix. Okay, so make sure you guys understand where that is, and I have a picture a little later in another slide that will kind of give you an idea of where that's located. Again, it's occurring to generate these NAD and FADH2, which will directly generate ATP in the electron transport chain. So if we have glucose coming in as pyruvate, and pyruvate entering the entering the mitochondria to generate ATP, it will first generate NADH and FADH2, and then later on in the electron transport chain, those carriers will help generate ATP. Same thing with fatty acids, same thing with proteins. They'll all be converted into this recognizable molecule. We will generate NADH, FADH2 through the Krebs cycle, and those will be used to generate ATP directly at the electron transport chain. And this is just basically further clarity on how important it is for acetyl-CoA to be used in the citric acid cycle, so in the Krebs cycle. So this is our key molecule in generating those electron acceptors. Here is another representation of what I just showed you guys. This is a linear representation though, so, and I have these bullets up here that we'll go through in a moment. So we talked about pyruvate, a 3-carbon molecule produced in glycolysis. So here's a cytosol. Pyruvate will enter the mitochondria. This is a enzyme complex that will help convert it to acetyl-CoA. So when pyruvate is converted into acetyl-CoA, which you can see here, acetyl-CoA is a 2-carbon molecule. In that process, we generate one NADH. Okay, so what is invested is up here. So the investment phase is all up top here, above the line. Okay, let me just quickly kind of go through the slide so that you know exactly what we're looking at. Here's what's invested. Okay, so these would be the oxidized forms of NADH+ and the oxidized form of FAD. And then there's an ADP here and a phosphate. And what this line represents, because some people had questions about this in class, this is just a linear representation of what is happening in this cycle here. All right, so I thought some people might appreciate this picture a little more. Okay, so again, we have pyruvate. It enters the mitochondria. It gets converted to acetyl-CoA. So we have a 3-carbon molecule that gets converted into a 2-carbon molecule, and we know that we lose a carbon because we can see it here being lost as CO2. Okay, we also gain one NADH. As the acetyl-CoA goes into the Krebs cycle, we basically start to generate these potential energy molecules here. So we have 3 NADH that is produced. Okay, that's really good, rich potential energy. We have 1 ADP that is converted to an ATP. And then we have 1 FAD, which is converted to an FADH. Okay, so when one pyruvate goes through this system, we generate all this robust potential energy. Okay, and this is just kind of like the most important part of understanding what is generated in the Krebs cycle. And this is just kind of reiteration, just telling you how important these things are in generating energy, okay, specifically in bioenergetics and metabolism. because they also can play a role in activating enzymes in the cell, which also can produce free radicals. So they can, FADH and NADH are great molecules for generating energy, but they can also be detrimental in activating enzymes in a disease condition that will be detrimental to the cell and the environment of the cell. So here is a little more detailed picture of what happens in the Krebs cycle. And I'm going to have a quick layout here of some carbon dynamics. So the C represents carbon, so we know that pyruvic acid, okay, or pyruvate, is a 3-carbon molecule, and we know that is converted to acetyl-CoA, which is a 2-carbon molecule. So we have pyruvate here. We have the conversion of pyruvate to acetyl-CoA, which is right here. You can see, just like on that other picture I showed you, we lose a CO2, so we lose a carbon, and now we have a 2-carbon molecule, which is the acetyl-CoA. Acetyl-CoA is now going to combine with oxaloacetate, which is this guy here. Okay, so oxaloacetate is a 4-carbon molecule, so you can see here that it's a 4-carbon molecule. And this is essentially going to, if you follow my cursor here, this is going to interact with acetyl-CoA, which is 2, and it's going to create citrate. And citrate will then be a 6-carbon molecule because we get 2 from acetyl-CoA combining with 4 from oxaloacetate, and that makes a 6-carbon molecule. Okay. Now the rest of all of this is unimportant. If you were in biochemistry, we would talk about all of this. All I want to really focus on is citrate all the way to oxaloacetate. Okay, so citrate will then be metabolized to oxaloacetate again. Okay, and that's why we call this a cycle, because this cycle will just kind of keep, as long as pyruvate and acetyl-CoA is coming in, this cycle will continue to just regenerate itself. Okay. In this process, we lose 2 CO2, and we'll talk about what sort of ramifications that has on the molecule. And most importantly is when citrate is generated, we can then generate a lot of this NADH that you saw on the previous slide. We have FADH being generated and NADH being generated here. So you can see that the oxidized form, NAD+, will become reduced in this process and produce these high energy electron carriers. Same thing happens here. Oxidized NAD+ gets reduced into NADH. The FAD, oxidized version of FAD, gets reduced into FADH2, and then again we have the same process where oxidized NADH gets reduced into NADH. I'm sorry, NAD+, that's a tongue twister, gets reduced into NADH. Okay. So to give you a little more of a visual on this process, I created this, and I hope it works. I'm going to quickly take a drink of water. Hang on. Okay. So pyruvate is a 3-carbon molecule. I put 3 little circles up here. And we know that when pyruvate gets converted into acetyl-CoA, we lose a carbon, and you can see that here. And then we're left with this 2-carbon molecule. Okay. If we look at oxaloacetate, we know that that's a 4-carbon molecule. 1, 2, 3, 4. If we follow this arrow here, oxaloacetate recognizes acetyl-CoA because of its carbon structure, and it's attracted to it. It wants to gravitate towards it. So it will basically, in this cycle, it will create a formation with acetyl-CoA, and now we have this 6-carbon molecule, okay, which is citrate. Now, citrate will be metabolized, and we will get 1 NADH, and we will get 1 CO2. Okay, so if we have a 6-carbon molecule and we have the loss of 1 CO2, then we're left with a 5-carbon molecule. Okay. This 5-carbon molecule will undergo further metabolism, further breakdown, will generate another NADH. And as you can see here, we lose another CO2. So what's going to happen? Well, this 5-carbon molecule is then going to turn into a 4-carbon molecule. And we will also generate 1 ATP. So I'm going to bring the 5-carbon molecule over here to the left. There it is. Okay. So we have this 5-carbon molecule. And of course, as I showed here, we lose 1 more CO2. All right. So what's going to happen is we lose another carbon, and we're left with this 4-carbon molecule. So, and as we identified earlier in this slide, this oxalacetate product is a 4-carbon molecule. So now, basically, this will enter the cycle again. So the next pyruvate that comes down will turn into acetyl-CoA. Oxalacetate, which is this 4-carbon molecule, will recognize it. It will bind and make a 6-carbon molecule, and around we go. It's also important to identify that through this process, we develop FADH, and then we develop NADH. Okay, so then this 4-carbon molecule is ready to go into the system again. Okay, so that's just kind of a visual to show you guys what is happening there using these actual carbon molecules. And I have no idea why the hell this one was over here. So that was supposed to be over here and drop here, but what are you going to do? Okay, next slide. So this is basically a reiteration of what I just showed you guys. It's almost the exact same set of slides, but I broke this down into a much simpler picture for you guys. And this I expect you guys to be able to draw on your exam. So we have acetyl-CoA, and you guys, that is a 2-carbon molecule, as I showed you on the previous slide. Here we would have oxalacetate, which is a 4-carbon molecule. We know that oxalacetate will bind to acetyl-CoA, creating a 6-carbon molecule. We will lose 1 CO2, which will create a 5-carbon molecule. In this process, we also gain an NADH. Okay, the 5-carbon molecule will get cleaved again. We'll lose a CO2, which will leave us with a 4-carbon molecule. In this process, we gain another NADH, and we gain a GTP, which is converted into ATP. And then from oxalacetate back to acetyl-CoA, we generate FADH2 and an NADH. Okay, so at this particular point here, we have oxalacetate again. Okay, so we started with 4-carbon. We met the 2-carbon to create a 6-carbon. The 6-carbon gets cleaved. The cleaving is necessary because that's what creates CO2 and also creates the electron carriers to basically accept the electrons and bring them to the electron transport chain. We have a cleavage again. We lose 1 carbon, so we have a 4-carbon molecule. That helps create more energy, more potential energy. We gain an ATP, and around we go. All right. So this part here you can read on your own. This is basically an overview of everything we've talked about so far from glycolysis to now. We can even look at this part of the cell, or I'm sorry, this would be the cell membrane, so we can even consider this the bloodstream up here, and you guys know everything on how insulin gets glucose into the cell. Once glucose gets into the cell, it's a 6-carbon molecule, right? We've talked about that. When glucose undergoes glycolysis, we create 2-pyruvate. Now, if we were exercising, this pyruvate can get converted into lactic acid. Let's just say that would happen over here, right, in this particular area here. And then that would be anaerobic glycolysis, because the glucose molecules that were produced, which is pyruvate, is being converted into lactic acid in an anaerobic capacity. So that means that no oxygen is present. We also know that through glycolysis we start to generate these high energy electrons, which are carried by NADH and FADH2. Those carriers will bring those electrons and those hydrogens down into the electron transport chain, but it will not do it directly. It will have to pass those hydrogens and electrons through those very highly regulated shuttling systems, like the malate-aspartate shuttling system. Okay. We know that pyruvate can also be metabolized aerobically, so that would be aerobic glycolysis. The pyruvate can go into the mitochondria. When it goes into the mitochondria, it gets converted into acetyl-CoA, right? That's that 2-carbon molecule. Here's that oxaloacetate here, so that's that 4-carbon molecule. These two will hook up, right? They'll do Netflix and cuddle and watch a movie and hook up. Right, isn't that what you guys say now? Like Netflix and chill. That's like the hookup lingo. So these two hook up. They watch Netflix and chill, and basically they create citrate, which is this 6-carbon molecule, and basically this will be cleaved back into another 4-carbon molecule, and this process will create more of these high energy electrons, which will be received and accepted by NAD plus and FAD, right? So when they accept those electrons, they get reduced to NADH and FADH2. So, and those carriers, if we follow this blue line, those carriers will also get down into the electron transport chain and donate them to create ATP. Okay, so that's where we kind of are now. So this is the big picture. So if you guys really want to study and really want to understand the concepts of everything we've been talking about, it's kind of all in this slide here. So if you walk yourself through this, you'll understand how all these things interact with one another to generate energy. Okay. So basically, now that we have talked about the Krebs cycle, and you know what glycolysis in the Krebs cycle does, now we're going to talk about the payoff. We're going to talk about why we have been focusing on these reducing agents, these coenzymes NADH and FADH2, why they're so important. So before that, I want to just quickly go over just a brief anatomy of the mitochondria. So the mitochondria is composed of 2 membranes. We have an outer membrane, which is represented here, if you follow my cursor. And then we have the intermembrane, which is here. Okay? And what I have in the intermembrane here is the electron transport chain. So we can see the protein complexes, we can see ATP synthase, and we'll go over all this in a moment. And then most important is this space between the outer membrane and the inner membrane. So this is the inner membrane space, and this is a very, very, very important space to help us generate ATP. So if we look at the big picture of the mitochondria, which kind of looks like a kidney bean cut in half, we can see the outer membrane here, okay? So, and that basically is represented here. And then we can see the inner membrane here, okay, which is represented by this image. And then we can see the matrix, okay? The matrix is this space inside, okay? So this would be the inner membrane, the outer membrane, and then in here is the matrix. And the matrix is where the magic happens, okay? So, and the matrix would be represented in this space here, where you can see the matrix. And look at what else is in the matrix. The Krebs cycle, or the citric acid cycle. So make sure you guys understand where these cycles are within the cell itself, or within the organelle itself. Okay. So I created this animation, and I hope this really helps drive home the message for what the electron transport chain does and how it works. So the electron transport chain is embedded in the inner mitochondrial membrane. Okay. So this would be the cytosol. This is where glycolysis is happening. This would be the outer membrane right here. This would be that inner membrane space. And this is the inner membrane, and of course the matrix. Okay, and the matrix is where the Krebs cycle is happening. So the electron transport chain is composed of 4 protein complexes, 1, 2, 3, 4. And it's also composed of two coenzymes that are mobile. They can move. So these are actually embedded. The protein complexes 1 through 4 are embedded in the membrane. They can't move. But these coenzymes, which is coenzyme Q and coenzyme cyto-C or cytochrome C, these are mobile and they can move within the membrane here, within the inner membrane. So let's direct our attention first to protein complex 1. So protein complex 1 interacts with NADH. Okay, so we can see that when NADH interacts with this complex, it becomes oxidized and its electrons are released. Oh, my phone's ringing. I'm going to ignore that. When its electrons are released, basically they are going to be processed through the protein complex 1. So you can see that protein complex 1 is going to process those electrons, and that is what starts to create what we call an electron current. Okay, so protein complex 1 is an electron acceptor. It's going to take the electrons, it's going to process it, and it's going to start creating this current. Now also, this complex 1 is also considered a proton pump. So the hydrogens that are associated with NADH are going to be pumped through this complex into the inner membrane space. And this is a very important process. So this is a proton pump. So when NADH interacts with protein complex 1, we have the removal of the electrons. The electrons is processed by this complex. We have hydrogen being pumped from the matrix into the inner membrane space. And that is step 1. Now, interestingly, if we look at this coenzyme Q, this is going to interact with the electrons that were processed by the hang on one second. Hang on. I gotta answer this. Hello? Hi. Are you here right now? Are you here now? Oh, okay. I'm actually doing a lecture online right now. Yeah, can I call you back in like 15 minutes? Okay. Okay, great. Thank you. Okay, bye-bye. Okay, I'm back. So let me try to figure out where I was. Of course, when I'm giving you guys a lecture, my phone never rings. Nobody ever wants to talk to me. And the one time I am trying to do a lecture with you guys, I get interrupted. Let me take my water here. Okay, let's get back to this. So what is important to understand, I could go back and edit that, but I'm not going to do it. So we're just going to keep this raw. What is important to understand is that the electrons here are going to interact with this coenzyme Q. Okay, so this complex 1 takes the electrons from NADH, processes the electrons, creates this current. The electrons are going to communicate with this coenzyme Q, and the hydrogen is going to be pumped up into the inner membrane space. Okay, so now what happens is this second complex, and this is kind of cool. This second complex only communicates with FADH. And what's interesting about this complex is it doesn't go through the entire membrane. If you look at complex 1, 3, and 4, those are all transmembrane protein complexes, which means they communicate with both sides of the membrane. So protein complex 1 can communicate with the inner membrane space, and it also has a section of it that is in the matrix. So it can make things happen on both sides of this membrane right here. On the protein complex 2, it only can communicate with the matrix. So it doesn't have a portion of this protein which can interact with the inner membrane space, which is kind of neat. So FADH2 is going to interact with protein complex 2. It's going to be oxidized, and basically what's going to happen is protein complex 2 is going to release the hydrogens from FADH, and those hydrogens are going to be stuck in the matrix, and that's a really important caveat to this system. Okay, it's also going to remove the hydrogens, just like we saw in protein complex 1, and it's going to pass these hydrogens through the protein complex 2, okay, further creating a greater electron current. Okay, and what's going to happen now is this guy, this Q, coenzyme Q, that picked up the electrons from the first protein complex, is now going to pick up the electrons from the second protein complex. All right, so the coenzyme Q is going to carry or transport the electrons. That's why we call this the electron transport chain. Okay, so it has the electrons, and it's going to now dump those electrons off at the cyto C location. Okay, so now it's going to communicate with this guy, and it's going to basically transport those electrons to cyto C, and now cyto C has the electrons. Now, it's very important to understand that this protein complex is not a protein pump or a proton pump. You see how this one here was transporting protons into the inner membrane space? This guy can't do it because this protein complex does not interact with the inner membrane space. So those hydrogens are left in the matrix. Okay, now with the cyto C that has the electrons from both protein pump 1 and protein 2, it is going to communicate with protein complex 4. And here it's going to unload the payload, which is the electrons. But before it does that, protein pump 3 is going to take these hydrogens, and it's going to put them into the inner membrane space. Okay, so 1 and 3 are protein pumps. 2 is not. 2 is generating the hydrogen so that these pumps can keep pulling hydrogen and putting them into the inner membrane space. Now, once we get to the protein complex 4, cyto C is going to drop off the electrons, and the electrons are going to go down the protein complex into the matrix. It's going to look like this. Now, in the matrix, we have molecular oxygen, and molecular oxygen is going to be attracted to these free electrons. And when the electrons and the molecular oxygen interact, we're going to have the creation of H2O. So essentially, water is a byproduct of ATP generation. Now, also, what is going to happen at this protein complex 4 is we're going to have another hydrogen or series of hydrogens being pumped into the intermembrane space. Now, what is so important about this gradient of hydrogens that we're generating and we're pumping into the intermembrane space is it's creating this environment that is very positively charged, okay, versus the matrix, which has less of a positive charge, okay? And this is going to play an important role in generating ATP itself, okay? So we have an excessively positively charged intermembrane space, and here we have less of a positive charge, which is going to create this gradient up here of hydrogens. Now, on this next picture, I'm going to show you it's the exact same thing. I have, I have the processes kind of spelled out a little bit, but we're going to add a new player here to this process, and that is this ATP synthase. Okay, ATP synthase is the last protein in this electron transport chain, which is going to directly generate ATP, and that's why all of these NADH and FADH have been so important, because they're contributing, you can see here and here, they're contributing to the development of ATP production in the mitochondria. So what happens is we have all these hydrogens that were pumped up into this space here. And these hydrogens are going to follow their concentration gradient down ATP synthase into the mitochondria matrix. I think one of them just animated by themselves. So what's going to happen is we have another hydrogen goes down, and then more hydrogens go down, and these hydrogens begin to basically turn this ATP synthase, which is, it's almost like a chamber. As the hydrogen goes down, it turns and it spins. And every time it turns and it spins, basically it helps regenerate ATP. So we'll have an ADP but get converted into an ATP. Okay? So this is the electron transport chain. Now, if I were to ask you a question on the exam, what would happen if complex 2 did not work? Let's say in the presence of free radicals, because a lot of free radicals are generated in the mitochondria. So with all these electrons floating around and all this oxygen floating around, there's a lot of opportunity for mispaired electrons and oxygen to become a radical. And when a radical is developed, it can destroy all these protein complexes. It can destroy the membrane here, so it can cause a lot of damage. So what would happen if if protein complex 2 malfunctioned and it didn't work? I want you guys to think about that, all right? Because I might ask you guys that question on an exam. Hint, hint, wink, wink. The next 3 slides are basically just summaries of what I just showed you on the electron transport chain. Okay, so I'm not going to read these to you because it's basically the exact same thing that I just told you, but you can read these and get a little more detail. So this slide, and the next slide are all just reiterations of the animation that I just showed you. To give you a better appreciation of I don't know why that went that way. To give you a better appreciation of ATP synthase, here's what it looks like, right? We have this positively charged intermembrane space because we created this proton gradient, right? And then in the matrix area, we have more negatively charged environment. And when these hydrogens go through ATP synthase, this rotates. You can see the arrow showing that it rotates. It looks like it's clockwise. And that rotation also allows this barrel to rotate, and that helps generate, take ADP and generate ATP. So that's just to give you an appreciation of how that enzyme works. So that is all I have for you guys. There are 3 more slides on this presentation that have to do with beta oxidation. And all I really want you to understand from beta oxidation is that when you have fatty acids, let me find the slide here. That would be this one. When you have fatty acids entering into the cell, okay, obviously we have triglycerides. Triglycerides are stored in muscle cells, but they're also stored in fat cells. And when triglycerides are broken down, we get glycerol and fatty acids, okay? Fatty acids, as I showed you, they go into the Krebs cycle as well, and they have to be converted into acetyl-CoA as well. Glycerol, we really don't generate a lot of energy from glycerol, so glycerol just kind of floats around in the blood. And protein is the same thing. Protein also gets broken down here in the Krebs cycle, which we had already mentioned. This would be that's a typo. This would be the beta oxidation process, and all you need to understand here is that when fatty acids are converted into acetyl-CoA, okay, they do, so that would be here, fatty acid acetyl-CoA, fatty acetyl-CoA, they generate the same sort of reducing agents that glycolysis did and that the Krebs cycle did with pyruvate. So that's all I want you to understand, is that fatty acids also go through beta oxidation in the Krebs cycle, and they generate FADH and NADH as well. Okay, that's the big take-home message. And that's it, guys. That's all I have for you. You are now caught up. I apologize by the interruption for the phone call. Because we were in the animation section, I'm not going to try to edit that whatsoever. I'm just going to leave it in there, and you guys can laugh at me, which is fine, because I laugh at myself often. Because if you can't laugh at you, who can you laugh at? So good luck this weekend studying. You guys have your review sessions with your TAs next week, and you also have your study guides. So that's it. Have a great day. Have a great weekend. Study hard.